for us to get started. So with this lecture, I want to cover a few topics that are somewhat related. In particular, we're going to discuss experimental design, quality control, and statistical considerations. Obviously, the title is for metaproteomics, but there's actually a lot of information in here that is very generalizable, both to proteomics, metabolomics, and frankly, most other omics studies. So it will be relatively general. My background is not statistics. We rely on it heavily, and we are informed by statisticians, and hopefully we can pass on a little bit of knowledge through some very good papers as well. So what I want to discuss today in this presentation is how you need to think about designing your sample set. What makes a good experimental design for metaproteomic studies? Discuss a little bit about quality control, and we'll talk about what that means, and then think about how your statistical analysis is supported by the prior two steps, the design of your study and the quality control procedures in use. I'm not going to reinvent the wheel. There's great papers out there that talk about this subject. And so I'm going to pull heavily on the statistical design from this paper from Oberg and Vitek, which was published several years ago now, but is a really nice overview on statistical considerations. It is more focused on proteomics, in particular clinical type proteomic studies, but the concepts are very transferable. So take a step back into omics generally. Omics studies enable you to have a very broad hypothesis. So you could define that broad hypothesis as I hypothesize that the proteome is changing between treatment and control conditions. You're defining what treatment and control are, and you're making the hypothesis that there will be differences. That is very generalizable and something that omics studies enable. Oftentimes, you also come with specific hypotheses. I hypothesize that bovine serum albumin can be found at higher concentration in cow plasma than rat plasma. Very intuitive hypothesis, but that's something that you can test through statistical analysis. And that is something that you can test within a proteome study. So you can look at all of the proteome between your rat plasma and your cow plasma, and one of those proteins that you detect is going to be bovine serum albumin. That is one small hypothesis within your larger experimental design, testing that big hypothesis. What this means, though, is that these omics studies are highly multivariate. And in a statistical term, what that means is that you run very high risk of false positive results. And so, as Tim mentioned in his talk, false discovery is something that we apply to peptide identification, protein identification. It's also something we have to worry about when we're doing our statistical analysis downstream. And so what we want is we want to set up our experiment and our statistical test to minimize the risk of those false positive discoveries. You don't really start thinking about this if you're doing one measure at a time. I'm looking at cholesterol in relation to heart disease, for example. That's one factor. We don't have to worry as much about false discovery because we're doing one statistical test. When we're looking at 10,000 proteins, the risk for false positives is high. If you generate random numbers 5 times out of 100, you're going to get a statistically significant result. And you can do this. This is an experiment that you can do. You can generate random numbers in R or SAS or whatever program you like and do stats on them, and you will get a significant t-test 5 times out of 100 on average. So that's what we have to be very mindful of. When we move to 10,000, that means we have a lot of false positive results that we're going to find by default. And you can visualize this. This is a wonderful figure I found on the internet. To summarize here, we've got type 1 error, which are false positives, where we have a dog claiming to be a cat. That would be a false positive claim. We have false negatives, which are type 2 error. I am not a cat, says the cat, which is incorrect. But really what we want to do is we want to hit all of the greens where the dog is saying, I am not a cat, our true negative, and we have our true positives where the cat is correctly claiming to be a cat. That's what we want our statistics to give us. And we get there in part through experimental design and appropriate stats. And they're really important in omics studies. So I just want to emphasize that very strongly up front. So let's take a step back and just overview what we mean by statistical inference or inferential statistics. And so basically the concept here is we're asking a question, does this group of subjects differ from that group of subjects? And we can't sample all of the subjects. So if we want to say that that cats are different than dogs, we can't sample every cat in the world. So what we like to do is we like to take a subsample of that large population. And that subsample is what we are performing our experiments on. And this is depicted graphically in that paper, where we have our two populations here, a healthy population and a diseased population. From that, we randomly sample from each population. And I highlighted random here because that's a very important point. If we randomly sample from these larger populations to generate the samples that we're actually performing our analysis on, then when we make statistical comparisons of our subsample, one against the other, then we can infer that it is representative of the full population. If this sample is non-random, then we are left with a bit of a conundrum. How far can we extrapolate back from our subsample to the full sample? And so you could think of this in a human population, for example. If you've got healthy population and a diseased population, but everybody's from Fort Collins, does that extrapolate to the wider world? Maybe and maybe not. But you can have far less confidence that it does than if you were to truly randomly sample from all of the healthy and diseased people in the world. So that random component to your sampling is a really important assumption that you are making when you're performing your statistical analysis. And therefore, you should be taking that into consideration when you're designing your experiment. What do you want your sample set to be representative of? Is it only representative of the Fort Collins population, or is it Colorado, or is it the United States, or is it the world? And that's a question you need to be thinking about in designing your experiments. And so in experimental design, this is nicely outlined as having two large goals. One is to avoid bias, where bias is a non-random source of error or a systematic error, which can be mistaken for a treatment defect. This is also referred sometimes in statistical terms as confounding design. And the second is that we want to ensure efficiency, where we minimize the random variation. And in doing so, we maximize our ability to detect all of the treatment effects that we're interested in detecting. And we can keep our study smaller by minimizing those random variant sources. This is achieved through three fundamental principles: replication, randomization, and blocking. And we're going to spend a little bit of time on each of these. So replication is looking at more than one individual. So if you have two groups, a healthy and a disease, and you see a difference in quantity, but your n equals 1 for each group, you can see that they are not the same. The question is that a meaningful difference. That could be twofold, that could be 1.5-fold, that could be tenfold. But we don't know whether it's a meaningful distance because we have no idea what the variability is in those. These could be randomly sampled from one distribution, and they would still look different. We just don't know how meaningful that difference is. Replication enables you to assign a statistical confidence to that level of difference. And so if we have 4 replicates of healthy, 4 replicates of disease, we can see that the distributions do in fact look different. You can perform a statistical test on the distribution of those replicates to say, yes, those are statistically meaningful. Alternatively, you can have high variance in your samples. All of the means for those plots are the same, but it's very clear that there is not a significant difference in our healthy versus disease on the right plot, where we have a lot of variance in our samples within our treatment groups. That means there's a lot of overlap in those distributions, which makes it less likely that this is a statistically meaningful difference that we're observing between those two means. So replication gives you confidence that any differences in means or medians or whatever your aggregate measure is in fact meaningful and reproducible. The second principle is randomization. Randomization guards against systematic bias. And these systematic biases can be they're displayed nicely here in one where you have on the left plot a sequential acquisition. And this is it's actually a really interesting example because it's actually people have made this mistake on some relatively high profile papers where they run all of the healthy samples on the first day and all of the disease samples on the second day, and you see a difference. And then the question becomes, is that difference due to the disease state, or is it due to the day I ran the samples on? And you don't know. There's a high opportunity for this being an artifact, statistically significant result. But the ultimate answer is we don't know. You can't tell the difference between those. So in this case, the day and the disease state are confounding. All of the treatments were run on one day, all of the controls on another day. That's bad practice. Randomizing, which is displayed in the next plot, helps you to eliminate that variation. So you now have treatment and controls run on 4 different days. And in fact, this is an example of blocking, which we'll come to next as well. And that's displayed here. So blocking is to say we now have, and this would be what is called a randomized complete block, where you have a treatment and a control within each day. And therefore your day serves as a statistical block. And now when you have implemented such a design, you can actually account for the day effect by saying day 1 on average is low, much lower than day 2. Day 2 is much lower than day 3. Day 3 is much lower than day 4. And then you can sort of normalize that away in your statistical model. You can account for that in your statistical model to give you the sensitivity to detect that increase between healthy and disease on each day.
Okay, thanks.
Of course.
So is this assuming that you're doing every single sample each day?
No, doesn't have to be. That would be, then in that case, you're replicating. You're replicating something, right? In this case, this is 3 different, let's say this is a true independent biological replication on day 1, day 2, day 3, and day 4. And so each of those 8 points is still a different sample.
Okay.
Yeah. But having that day effect, because we have some concern, at least, if not confidence, that a day effect may confound our results. It may be a significant systematic source of variation. By including both treatments and controls, either in a very rigorous manner, a healthy and a disease in each case, or randomly assigning them, it enables us to account for the day effect. So now let's talk about N. This is something that always comes up when talking about, just got an email yesterday about N, the number of samples to run. How many replicates do you need? And the answer is more is generally better, but there's a balance. And so the best way to approach this is to use what's called a power calculator. And what this does is it says, you make some assumptions. You say, I want to be able to detect a 2-fold increase, and I expect my variance to be this. How many replicates do I need to have this much confidence in my result that I'm going to return all the true positive results? And there's a statistical approach that you can use called a power calculator. You can find them on the internet. You can also talk to your local statistician. And we have a wonderful resource here on campus, the Stats Lab, the Graybill Statistics Laboratory, where there are experts designed to help everybody on campus with their statistical questions. I highly encourage you to use them. I do. More than they're probably happy with at times. And so there are resources for that. Now, that said, you can't do stats if you have less than 3. I'm always going to tell you the absolute minimum is 3. And then I'm going to tell you that there's a decent chance that one of your samples looks weird and you're going to want to consider it an outlier. And if you throw that one away, you can't do stats. So try to round up to 4. But usually you want to be higher than that if you can, especially in these omics experiments. The more replicates you have, the more confidence you have in your statistical results. That raises the question as to what is a replicate, and this is an important one too. There are different types of replication. Analytical replicates are not the same as biological replicates. We can't take your one sample, run it 3 times, and do stats on it. Remember our inference step on that first slide. If we are randomly sampling 1 microliter from your vial, what do we want to extrapolate back to? Those 2 samples, right? We're randomly sampling the prep that you have done on these two samples, that is not a biological rep. So we can't say anything with those statistical analysis about the biology that you're interested in studying. A preparation replicate, if we take two subsamples from your sample and prepare them independently, that is still not a biological rep. A biological replicate has to be two independent samples from whatever your study is, whatever your sample type is, each prepared and analyzed independently. And then we have to define what independent means. And this can get, there's a lot of gray area here. And you always have to be thinking about what conclusions am I trying to support? Do I want to compare the human population or do I want to compare the Fort Collins population? If we're talking about independence, are two plates of microbes from the same stock culture independent? Anybody have opinions? Not so much, maybe. But if this is one species or one strain collected from this location, and that's one strain collected from that location, and you developed one isolate, that's all you've got. So there are times where that's what you have to use. You just have to keep in mind, what can I infer from the statistical results that I generate? How about two tissue biopsies from the same liver? From the same person, right? So one might consider the person a block, even. Like there's ways to statistically manage that. Are two leaf samples from the same plant independent? There's developmental differences. This might be an old leaf, that's a young leaf, but they're still from the same plant, right? And it all depends on what you're trying to extrapolate to. So you always have to be thinking about that as you're designing your experiment. What conclusions do I want to generate from my statistical results? So quick summary of that, and I encourage you to read the paper. That's a great paper that's out there that organizes this. I pulled their figures, I used their concepts. Omics data are very sensitive to experimental design flaws. You want to be careful in designing your experiment. And that's because they're so multivariate or multifactorial. We've got multivariate. We've got thousands or tens of thousands of variables that we're looking for. Random number generators can generate significant results. Proper design prevents these artifactual statistical results, and we need to rely heavily on replication, randomization, and blocking to make sure that the data that we are reporting on is of high quality and we're not introducing bias or high variance, which masks treatment effects. And I just wanted to put an aside here. This was there was a great scientist, I think he's at Rice, if I recall, named Keith Baggerly. And about a decade ago, he gave a talk at the mass spectrometry conference. And I love watching his talk. So he's a statistician. And he actually went back and looked at some of the early cancer biomarker studies using MALDI, time of flight, mass spectrometry. And he performed what he called forensic statistics to try to understand what they did and actually replicate just the data analysis component, and was able to actually find that some of these studies are pretty highly flawed in some of the ways that we've talked about, and gives a great talk on the importance of this. And if for nothing else, watch it so you can convince yourself that you do not want to be the subject of a Keith Baggerly talk. So do your job to prevent becoming a Keith Baggerly subject. All right, we're going to move on to quality control. Before we get there, any questions or thoughts on the statistics component, experimental design?
Just wanted to confirm that I got the question that you had asked initially, like what we want our sample set to be representative of is the place that we should start.
I think it's always worth thinking about that, right? So you have your samples sometimes. You're given a sample set, and the question is, you work on sourdough, and you've got two different starter cultures, and you're trying to compare them. Are you trying to extrapolate microbial composition? Are you trying to extrapolate to all the sourdough everywhere, or are these considered representative? How did they come about? Is this random or non-random? And those sorts of questions should guide your design to the extent that you can, right? Sometimes you don't have much option there. But at the end of the day, it should at least guide your interpretation of your results to say, what are my samples representative of? And to prevent you from overinterpreting it. Quality control is a separate but related subject. And this is an important subject that is often neglected. And what it is the ability to understand your sources of variation and potentially control for them. And there's another paper that I will pull some information from here. And I want to give all credit where it's due. So I've got 3 papers I'm pulling from. I encourage you to read them all. Quality control is defined as a systematic approach to, which makes it possible to objectively, and I highlighted objectively, assess the quality of a mass spectrometry experiment. And I highlight objectively because this is something where you are trying to come up with a way to convince the world that your data is of high quality. That's what quality control should enable. Because if you can't demonstrate that, then the readers, the audience, whatever that audience is going to have questions about the validity of your experiments. And so that objective assessment is you can think of it as a scientific norm or standard that you are going to put out into the world to say this data set is good. And that most readers will accept that if you put these metrics out there, they will agree with you. And so this demonstration of data quality could include things like instrument sensitivity, analytical or preparation, reproducibility, confidence in annotation. We talked about false discovery rate on peptide identifications. You want to be able to demonstrate through your quality control approach that some of these things have been accounted for, and we've got metrics that can convince the audience that they're good. So let's think about all of the sources of variance that we could potentially include in a proteomics or metaproteomics experiment. So it's basically any step in the process, literally any step in the process. And what we're trying to do is to maximize the sensitivity to the experimental design induced variance. So if we have a treatment and a control and they're different, we want to make sure that we haven't introduced so much variance that their distributions overlap. We want to be able to detect real differences. So to do that, we have to minimize all of the non-biological, ecological, environmental sources of variance that we can to detect the one that we're interested in, the treatment. These quality control procedures help because they enable us to recognize variance as it's occurring, potentially, and possibly to recognize the source of that variance if we have taken all of the proper steps to think about that. So these quality controls can also at times be used to correct for undesirable variance. And so if you recognize that your instrument sensitivity is declining by the 300th sample that you've injected, there are ways of correcting for that to a point. And there's a nice figure from the paper that sort of outlines in a proteomics experiment some of the sources of variation that we might be able to recognize in our proteomic analysis. That includes variance from sample preparation, digestion efficiency, and modifications, variable modifications in particular, as Tim had pointed out in our prior talk. You can have variance induced by the liquid chromatography itself, injection carryover. So if I make an injection in one sample and then I inject a blank, do I see peptides from the prior injection? That can happen. It's a real technical artifact, and it's something that can be very small and trivial, or it can be large. We want to make sure it's not large. So blanks are very useful. Column lifetime, we can get degradation of peak shape and separation over time as the column ages. We can have variance from the mass spectrometer, which might include the dynamic range changing, that we don't see responses to increasing concentration, or in ionization efficiency, that little tip where we saw the spray coming out of yesterday, for those of you that went to the lab, it ages. And if our spray becomes less effective, we get less signal, and that can be introducing artifacts. We can also introduce artifacts through the informatics if we're not careful. That's probably a little bit more rare. Most of those are going to be a little bit more random, but it's certainly worth considering the artifacts that you can potentially introduce, or the variants you can introduce. There's a lot of different ways people have approached quality control in proteomics experiments, and we'll talk about some of these in just in an outline form. Broadly speaking, you can break this into 2 big classes for controls: external standards and internal standards. An external standard is a different sample than your sample. It's a different injection than your sample set at all. So you might come to us with a metaproteome sample, and Gustavo and Dora, when they set up the sequence, will run something called HeLa, HeLa cell protein extract. It's a human cell line. It has nothing to do with your sample set, but it is a standard sample that the proteomics community has adopted widely as sort of a quality control type sample. And we will use that to see how is our instrument behaving. So it's a separate sample completely. Another one could sometimes be a single protein. Bovine serum albumin, BSA, is a common protein standard for a lot of people. You can look at things like how much of the protein do I cover when I make an injection of the sample? How many of the peptides do I detect? So our complex sample could be something like HeLa. Yeast is another one that's commonly used. E. coli is another one. They mentioned in the paper this Paracoxus. I don't even know what that is, but it's used and mentioned, so I brought it up. More rarely, people will actually use a mixture. So they'll say, I'm going to take some HeLa, I'm going to take some yeast, I'm going to take some E. coli, and I'm going to mix it all together. And that then enables you to actually report on your ability to quantify because you're mixing in known ratios. And you can actually be testing, does my assay report on quantitative change that I know to be there? And so there's ways of implementing quality controls that enable you to test quantitative response as well. Oppose external standards to internal standards. Internal standard is something that you actually spike into your samples. So if you come with a metaproteome sample, you can spike internal standard peptides into that. The benefit of the internal standard is that every injection, including your samples, now has a quality control signal or signals in it. And so if you have a single bad injection on your sample, that should be recognizable by the internal standard approach. But the external standard approach would not report on that. The downside of his internal standards is that they're often relatively simple, that you can't spike in 10,000 internal standards to make it representative of all of the peptides in your sample. That is economically not feasible, and it would cause your data quality to actually go down because there's a lot more signals that are now competing with your signals. And so this might be 1 to 50 peptides that you would spike in. And when you spike them in, these are going to typically be required that these are isotopically labeled, or at least you have a high degree of confidence that an unlabeled peptide isn't present in your sample. So you have to make some relatively informed decisions about what peptides you're spiking in, because you don't want them to be confused with your sample peptides. Another type of quality control is a blank. As I mentioned, this can be really useful for recognizing things like liquid chromatography carryover. If I inject HeLa and then I inject a blank and I detect human proteins that are present in HeLa in my blank, I know that there's carryover. And then you can actually look at it and say, what is the level of carryover? Does it look like 50% of the signal that I injected last time? That would be very bad. Or does it look like 0.5%, which is more typical and not necessarily worthy of any instrument issues or maintenance? Ideally, your QC approach, your QC sample type reflects the properties of the sample under analysis. And this is a really difficult ideal to achieve, particularly when you're talking about metaproteome samples. The complexity there is so high. It's nearly impossible to come up with a good QC sample that's representative. We do use some approaches. There's a commercial product from Zymo that we use, and I think some of you actually prepared it in the hands-on portion, which is a mixture of species. I think we have one that's a 12 species mix, if I recall, of microbial species, proteins, and peptides. An oral microbiome they just came out with. And so there are samples of pretty good complexity of known composition that we can use for quality control in these types of studies. Now, in practice, we need to figure out how to use these QC samples, and there's different ways of doing it. You can see going from top to bottom in the figure. The blue samples are QC samples. The black samples are your genuine biological or ecological samples. And you can see the frequency differs, the spacing differs. The benefit of injecting rarely is that you run less risk of contaminating your samples with the QC samples, that you have a shorter experiment. If you've got 20 samples and you run only two QCs, it becomes 22 samples in total. So it reduces the cost a little bit. But as you go to increased frequency going down and including more types of QCs, you gain more information from your quality control process. And that can be really valuable in diagnosing the problem. So some people will regularly intersperse them, and this might be every 5 injections or every 15 injections. And that's something that you need to be thinking about when you're putting together the acquisition list. Any thoughts on this? Now, when we're getting to the quality control side of things, and we need to think about how do we objectively report on data quality, there's a lot of different things that you could be reporting on. I'm not sure that there's a consensus that one of these is more important than the next, or that one of them is absolutely critical and the others are optional. I think the best approach is to be thinking about as many of these as you can with the resources that you have available. So one of them that we can be talking about is the number of artifact modifications. So we mentioned those variable modifications. These typically come from sample handling. If we can look at all of these and we say that, well, some of the samples have extremely high levels of this and others don't, that might be a cause for concern, right? And what you do with that information is something that we need to figure out. But recognizing that there might be a bias imparted through the sample handling is an important first step. We can also be looking at digestion efficiency. We can look at that by looking at how many miscleavages do we see. If we see relatively low miscleavages all the way across the experiment, we know we're good. If we see low miscleavage in the controls but high in the treatment, that's a problem. That's another confounding variable, and it's pointing us to a sample prep digestion problem. Sample loss, how much peptide or protein do we recover from a given amount of tissue? Contaminants, Tim actually mentioned a database. I don't remember exactly what he called it. He gave it the nice name. It's actually, if you look at the bolded letters, CRAP, it's commonly referred to as the crapome. And these are proteins that are common contaminants in proteomics experiments. And that is something that we can be looking at on a sample by sample basis once you've done your analysis. Chromatography, does our peak shape or width change over the course of the experiment? Retention time, how much does that drift? Carryover, if we've used blanks, can we be looking at what percentage of HeLa or sample signal is in the blank in the next injection? Those are things that can be quantified. Mass spec, what is the signal intensity? And mass spec is a device where the sample goes into the instrument. It's not like a UV where it's trapped in a cuvette. It goes into the instrument. What that means is it actually dirties the instrument over time. So mass spec is sensitive to signal drift. It's something we have to be mindful of. Sequence coverage. If we see the same protein in every sample, do we get about the same sequence coverage through the entire experiment, or does it drop? That might, again, be reflective of signal intensity drift. Mass accuracy. The instrument isn't perfectly stable in how it measures mass. And if we have high mass accuracy at the end of the experiment, then at the beginning, that's a problem. And we can look at total protein identifications or peptide identifications and report on that as well. So these are all metrics that you can be reporting on from your experiment, depending on which level of quality control sample you have included. But those are all potentially reportable. Values like this, in particular, if you're using internal standard peptides, you can be looking at mass accuracy on a peptide and sample-specific basis and say, yes, it is consistent. I'm getting 5 ppm mass accuracy or less in every sample. And that gives you a lot of confidence that the mass spec is behaving well. So very quick summary of that section. Quality control is a really important component of your experimental design. I wouldn't consider it independent of your design. It's your experimental design is what you think about with the biology. The assay that we run on the instrument and your sample prep, if you're doing your sample prep, is a component of the experimental design and should incorporate quality control type thoughts. The goal being to objectively demonstrate data quality at the end of the day. When you go to publish this, whether it be a peer-reviewed journal or a report to whoever has paid for the study, you want to make sure that you can convince that reader, that audience, that this is a high-quality data set. That's what your quality control should enable. And just as importantly, with good quality control, you can more confidently interpret your results. Now we're going to touch a little bit on the analysis of proteomics data. And this is going to be the most ambiguous portion of the talk because statistics is a massive field on its own, and there's no way that we can cover it. But we'll, again, reference a paper that has tried to summarize some of the tools that people use in proteomics studies. So when we're going to the analysis of proteomics or metaproteomics data, the questions that we might be thinking about are which proteins are responding to my treatment. That is, which proteins are higher in my disease versus control, in my healthy soil versus my burned soil. What pathways? So Tim mentioned mapping to KEGG. What biochemical functions? We can be thinking about things like GO terms, gene ontology terms. Which taxa are present in a metaproteomics experiment? How do these responsive proteins relate to each other? Again, thinking about pathways or biochemical interactions. And there's a lot of studies now that are using multi-omic approaches, where you not only have your proteome or metaproteome, but you also have your metagenome, your metatranscriptome, your metametabolome, your metalipidome. And you can be correlating these things to each other, given the state of your experiment and the experimental design and what data you have. Those can be quite complex, but it's a relatively frequently applied approach these days as the cost of many of these omics drops. And so this paper gives some relatively big picture overview figures, and I just want to highlight a couple of these. First thing you get from these metaproteomics and proteomics experiments is a peptide inventory, right? You can say this peptide was or was not detected. That doesn't necessarily mean that this peptide was absent if it wasn't detected, but you can at least say it wasn't detected. You can then be doing analysis at the peptide level on by performing things like gene coverage studies. You can be looking at which peptides are the highest responders, which peptides have the most signal. And you can also be doing things like motif analysis. So is there a particular, if I look at all of the significantly changing peptides, is there a common motif in there that might indicate a particular genetic control element, for example? You can also be doing analysis on the protein level. You can be looking at things like physicochemical properties. So are we just detecting soluble proteins, or do we get a bunch of membrane proteins too? Is it more acidic proteins on average than, and that might give you some insight into the biology, depending on the type of study that you're doing. Once you've mapped your proteins to gene ontology type functions, you can actually be looking at your statistically significant proteins and saying, are specific GO terms changing more than another? This can also be done with these PFAM domains. So again, sort of regions of a protein that might represent certain biochemical functions in a little bit more sequence-specific manner. And you can also do things like network mapping. This could be mapping to biochemical pathways, but there's also tools such as String, for example, that will map proteins together based on known biochemical interactions. Got two metabolic enzymes that actually bind to each other in solution and are known to interact to form a little metabolic functional unit. And that is captured in a database. There are tools that will put those proteins in your proteome data set together. So you can see these things in the same context that they're existing biologically. At least that's the inference. The goal with these statistical analysis is always bring data to biology. So one of the things that people will commonly do is look at the fold change analysis of individual proteins. And so this prompted the question that I asked Tim, are you doing this at the subgroup or the group level, at the protein? You could also be doing this at the peptide level. Some argue that we should be doing a lot of stats at the peptide level in these types of studies. But you can be doing fold change analysis. Often this is going to be coupled to something like an analysis of variance or a t-test. So you can be looking at your replicates and saying, yes, I have a significant p-value. These things are meaningfully different. It's really important to remember that randomly generated data will generate significant results. This will happen. So do false discovery rate correction. We talked about false discovery in protein ID. We also have to talk about false discovery in statistics. It does happen. It will happen, even with randomly generated data. So if you do not do false discovery rate correction, there's a very good chance that the story that you tell about your data isn't right. You obviously wouldn't be telling true stories. And so FDR correction helps enable that. There's a lot of studies that will use time course data. There's special statistical tools that you can apply in this case. If it's, one or two time points, then it's very similar to a standard study. But if you've got 10 time points, then you need to be thinking about specific time course type statistical analysis. That might include repeated measures, curve fitting. Ultimately, you can always find some good precedent in the literature. And this is always a good idea to do. Find a good example paper in a reputable journal that has passed peer review. And if you try to mimic those statistical analysis, you're going to be much more likely to fly through your peer review process than if you try to develop something completely novel. There are times when novel is what you want. Talk to your statisticians about that. And I do provide a link here to the stats slide. There are also multivariate methods. What I talked about in the prior slides was mostly univariate. We're testing one peptide or one protein at a time. Oftentimes, we want to ask, does the proteome change? That's a multivariate question. And there are specific statistical tools for that. You'll often see these plots like you see on the top right here where the heat maps. That is often the product of hierarchical clustering analysis. It doesn't have to be, but oftentimes those heat maps are clustered. You'll see the little trees on the right and on the top, and that is clustering analysis. This is an unsupervised technique, which means this hierarchical clustering algorithm doesn't know your treatment and your control samples. It's just grouping based on the pattern of quantitative variability. And what that enables is pattern visualization, really. Principal component is another multivariate approach, also unsupervised, that enables you to visualize patterns. That will look something like that plot on the bottom. It's not really a statistical test. It just enables you to look at your data, which is extremely complex, in a nice, concise figure. Which can be very valuable. This is a supervised approach, PLSDA, partial least squares discriminant analysis, where your statistical tool actually knows your treatment and your control and is trying to separate them. These types of approaches can be very prone to false discovery as well. You can generate random data. You give random data to PLSDA and say there's 5 treatments and 5 controls randomly assigned. It will separate them. And there's tools that you can use, such as bootstrapping, subsampling, leave one out, cross-validation, that will enable you to increase the rigor of that study. So make sure that you're using the appropriate controls even within this, especially when you're in a supervised multivariate tool. So I just wanted to throw in a couple of examples here of what people are doing with recent proteome and metaproteome type studies. So here's an example where they took proteome data and they're actually correlating it to genomes. And so they've got in this study thousands of genomes, human genomes, thousands of proteomes, thousands of health records. And through statistical analysis, they can start linking all of these things. Hundreds of thousands of associations that turn up, with proteomics being a key component of that analysis. Here's an example of temporal variations where they're looking for markers, in this case, a proteomics study for Alzheimer's disease. And actually through the design of the experiment can determine that there are protein biomarkers that show up often 2 decades before clinical onset of the disease. And so this is another time type study, a temporal type study, where you had to be taking into account, you can just barely see on the tiny font on the bottom of that figure, the estimated year of disease onset is that big black line going up and down. And so by incorporating that time component, they can see that not only these are good protein biomarkers of current disease, but actually potentially prognostic, showing the development of the disease in advance of the clinical symptoms. And here's an example from a metaproteomics study where they're actually looking at these metaproteome samples to look at something called NDAMO, nitrate-driven anaerobic methane oxidation, and trying to infer which taxa are performing the biochemical reaction that they're most interested in. And so they developed a data set, a sample set, and quantified nitrate-driven anaerobic methane oxidation, and then associated the metaproteome data with it. And in doing so, they were able to, with a fairly high degree of confidence, narrow it down to these taxa that seemed to correlate the most with the biochemical reaction that they were interested in. And so this is actually a figure from Unipept, which Tim had showed us before, and which those of you that are participating in the hands-on will actually be using yourselves. So just some examples. Quick summary. All omics, proteomics and metaproteomics included, benefit from good experimental design. You need to be thinking about your experimental design, including replication, randomization, and blocking to ensure that your data is going to generate good results. Quality control is something you should be thinking about before the experiment and evaluating after. Make sure that you can report objective metrics of data quality. And finally, good design and good QC make for stronger statistics at the end. If you want to detect those real changes, replication, randomization, quality control, all of those things make your statistics more sensitive in picking up those true positives and minimizing those false negatives. More information can be found. I will just throw up the links again and acknowledgements. So any questions, comments? A lot of coverage, a lot of talk about. Really a prompt to just make sure that you're thinking about these things at the various stages of your experiment.